Hello students, today I'll be talking about lipid metabolism, and in this lecture I'll be covering saturated and unsaturated fatty acids, oxidation of fatty acids, in which I'll be specifically talking about beta oxidation. Now to start with, what are lipids? The word lipid comes from the Greek word lipos, which means fats. So basically, lipids are a diverse group of heterogeneous compounds related to the fatty acids, including fats, oils, waxes, and smaller compounds like enzyme cofactors, vitamins, electron carriers, hormones, intracellular signal molecules, etc. And they are so diverse that their only common defining feature is their insolubility in water, which means all of them are hydrophobic in nature. Therefore, as a general characteristic, they are relatively insoluble in water and considerably soluble in organic solvents like ether, chloroform, benzene, etc. Lipids are important constituent of our diet because of their high energy value and also because of their role as solvents for many important fat-soluble vitamins in our foodstuffs. Now, this diverse group of heterogeneous compounds called lipids can be grouped into three main categories. The first category is called the storage lipids. The second category we will call the membrane lipids, and the third category comprises of the other lipids. Now, these storage lipids, the first category, comprises of all types of natural fats like the palm oil, vegetable oils and other plant oils, waxes, glycerol ethers, and other derived oils. The membrane lipids, the second category, comprises of the phospholipids, the glycolipids, the sphingolipids, and the steroidal lipids. These membrane lipids, they act as the membrane backbone in the biological membrane structure. As you might remember, the lipid structure in a fluid mosaic model of the cell membrane. The other lipids comprises of the enzyme cofactors like the vitamin E, vitamin K, quinones, and the pigments like the carotenoids, the hormones like the testosterones, and the signal molecules, and other vitamins like vitamin A, vitamin D, etc. Now, if we look at the structure of the fatty acids, they are long-chain organic acids made up of 4 to 30 carbon chain backbone containing a single carbonyl group as a head and a long non-polar hydrocarbon tail. This long non-polar hydrocarbon tail gives them their characteristic hydrophobic and greasy nature. The fatty acids do not occur in uncombined state in cells or tissues, but are present in covalently bonded compounds in different classes of lipids. They are stored as energy reserves in tissues. The fatty acids which occur in natural fats are monocarboxylic, which means they contain a single carbonyl group and contain an even number of carbon atoms. Now, what are saturated and unsaturated fatty acids? Basically, saturated fatty acids contain only single bonds between the long carbon chains, and unsaturated fatty acids contains at least one or more double bonds between the long chain carbon backbone. The fatty acids are stored as an energy reserve in the form of fats in animal tissues through an ester linkage to glycerol to form triglycerides. Now, if we look at them separately, the saturated fatty acids have the general formula of CnH2n plus 1 COOH. And if we look at the carbon atoms, mostly they are even numbered, straight chain fatty acids, and very rarely odd numbered, found in both plants and animals. In animal fats, the palmitic acid and stearic acid containing 16 carbons and 18 carbons respectively are the most abundant, example in milk fats, body fats, etc. The unsaturated fatty acids comes with the general formula of CnH2n minus X COOH, in which the X is any odd number. And the unsaturated fatty acids are classified on the basis of the degree of their unsaturation, which means the number of double bonds contained between the carbon atoms in the backbone. So the unsaturated fatty acids are of the two types: monounsaturated fatty acids, in short called MUFA, and polyunsaturated fatty acids, in short called PUFA. The monounsaturated fatty acids, MUFA, as the name suggests, contains only one double bond, an example of which is monoethanoic acid with the general formula of CnH2n-1 COOH. The polyunsaturated fatty acids contains more than one double bond. There are three types. The diethanoic acids contain two double bonds, which in common terms are called linoleic acids. They come with the general formula of CnH2n-3 COOH. And the second type, triethanoic acid, contains three double bonds. They are commonly called linolenic acid. They come with the general formula of CnH2n-5 COOH. And the third one, tetraethanoic acid, contains four double bonds. They are commonly called arachidonic acid, and they come with the general formula of CnH2n-7 COOH. So basically, in most unsaturated fatty acids in plants, the primary double bond is between the 9th carbon and the 10th carbon, which is designated as delta 9. If there are additional double bonds, they are found between the delta 9 and the methyl terminal end of the fatty acid chain. In mammals, the polyunsaturated fatty acids can have up to 22 carbon atoms and 6 double bonds. And in plants, the polyunsaturated fatty acids do not exceed 18 carbon atoms and 4 double bonds. Now these linoleic acids are commonly called lin fats, and the linolenic acids are called lan fats. The linoleic, linolenic, and arachidonic acids are essential for the normal functioning, but they cannot be synthesized by the cell themselves, and they have to be obtained from the diet. Hence, they are called essential fatty acids, or EFA. Now, let's talk a little more about the lin and lan fats. The lin fats, along with longer chain fatty acids derived from it, are referred to as N6 fatty acids, wherein the first double bond occurs on the 6th number carbon from the methyl end of the fatty acid chain. Similarly, lan and its derivative fatty acids are called N3 fatty acids. Vegetable oils contain two types of polyunsaturated fatty acids: lin fats with 2 double bonds and lan fats with 3 double bonds. Lin fat is more abundant in nature and is present in all vegetable oils, whereas lan is present only in some vegetable oils such as mustard oil, rapeseed oil, and soybean. Vanaspati are hydrogen oils manufactured from vegetable oils by the process of hydrogenation. So basically it is a manufactured saturated fats, and although derived from vegetable oils, which are basically unsaturated, they behave like other saturated fats of animal origin. Fish oils are good sources of both lean and lan fats, hence considered very rich nutritionally. All foods contain small quantities of bound fat or invisible fat. Example, in green leafy vegetables and pulses, they are rich sources of N3 fatty acids. Now, we must remember that too much N6 fatty acids or the lean fatty acids with very little N3 fatty acids or lan fats in our diet is undesirable. So, a good combination of lean and lan fats in our diet is good and is desirable. Now, before we move on to the beta oxidation of fatty acids, let me briefly give you an idea about the fatty acid biosynthesis. Or in other words, we will be discussing exactly from where does the fatty acids come into our body. Now, in our body, the fatty acids are accumulated as triglycerides or TG from ingested food. Also, they are accumulated as triglycerides resynthesized from glucose and protein metabolism, which we call the biosynthesis pathway. As you must remember, in the glucose metabolism, after the molecules of glucose are broken down into pyruvate, by the process of glycolysis, this pyruvate enters the TCA cycle in the form of acetyl coenzyme A. This acetyl coenzyme A, in fact, is degraded from pyruvate by an enzyme complex called the pyruvate dehydrogenase complex. Now, this acetyl coenzyme A, when it enters the TCA cycle, the first stable compound it forms is the citrate. This citrate can also be transported out from the mitochondrial matrix into the cell cytosol by certain shuttle molecules called the citrate shuttle. The enzyme citrate lyase helps in this whole process, and after the citrate is transported back out into the cytosol, it forms acetyl coenzyme A. This acetyl coenzyme A is acted upon by the enzyme acetyl coenzyme A carboxylase to form malonyl coenzyme A. This malonyl coenzyme A is acted upon by fatty acid synthase enzyme to form palmitate, which is a 16-carbon compound. This palmitate is the most abundant fatty acid in animals.